Our nearest planetary neighbor in space is the Moon. This object of poetry and speculation for countless generations gave rise to the concept that the Earth was only one world of many. As early as the 5th century BC, a Greek philosopher expressed his opinion that the Moon was an Earth-like world. About 165 AD, a Greek writer named Lucian of Samosata wrote a true history, an account of a trip to the Moon. The hero was carried to the Moon by a whirlwind. Over the years, other stories about moon journeys have been written as well. While astronauts were being trained for the lunar landing, and the many parts of the spacecraft were being tested in laboratories and shops, there was one other facility that was essential to the Apollo program.
Of major importance to the scientific study of the material from the Moon will be the Lunar Receiving Laboratory located at the Manned Spacecraft Center in Houston. This laboratory will dominate our attention immediately following man's flight to the Moon. Here, first to an almost totally bare room, the astronauts, their spacecraft, and equipment will come upon their return. Meanwhile, work with the lunar materials, which will weigh a total of perhaps 50 pounds, will have begun. And seldom will an equal amount of any other material receive as much attention. Collected with geologic hand tools designed especially for use on the lunar surface. The materials will be packaged in aluminum containers. These are designed to maintain inside the vacuum of space and to prevent contamination from the Earth's environment. After unpackaging operations in ultra-clean vacuum chambers, the samples must be proved biologically safe. This will be done in a series of tests, using animals kept in germ-free surroundings as well as in normal environments. Other forms of life, for example, plant tissues and eggs, will also be used in the biological tests. Many of the biological safety tests will take place in cabinets equipped with arm glove assemblies. The cabinets enclose carefully regulated environments. Meanwhile, the scientific work will have already begun. As soon as the material samples are received, sorted, and classified, scientists begin preliminary investigations. They will study the physical, chemical, and mineralogical properties. They will analyze the minute traces of gases emitted by inorganic and rare matter. And by possible organic matter. They will measure radioactive characteristics, a job which must be undertaken as rapidly as possible to prevent loss of short-lived data. For this, there is a heavily shielded facility located 50 feet underground to help prevent interference from natural outside radiation. In many ways, the Lunar Receiving Laboratory will serve as a major tool for the scientific investigations of material from the Moon. But the work here is only the beginning. Once preliminary investigations have been completed and samples have been classified and prepared for delivery, they will be distributed to more than 100 scientists. In laboratories throughout this country, in Europe, in Canada, in Japan, answers to basic questions of science will begin to come forth.
July 20th, 1969 is a date that is recorded forever in the chronicles of history. For on that date, visitors from Earth stepped onto the Moon's surface for the first time. They were to conduct experiments and return samples of lunar rocks to Earth. During the Apollo era, 840 pounds of lunar rocks and soils were returned to Earth. Did these lunar materials contain evidence of life on the Moon? In this program, we will see the vast amount of geological and botanical studies done with the lunar samples of Apollo.
In Apollo 11, man's first landing on the moon, there was no objective more important to science than the collection and return of samples of the lunar surface. Never before had we had the opportunity to examine extraterrestrial material from a positively known location and context. Fortunately, at the site of the landing, in the southwestern part of the Sea of Tranquility, there was a considerable variety of lunar material, much of it debris from nearby impact craters. At first, in case of a contingency, samples were chosen and collected rather rapidly. Later, as operations continued to proceed smoothly, samples were chosen and collected more deliberately. Within 5 days after the samples were picked up on the lunar surface, where they had lain for millions of years, they were delivered to the Lunar Receiving Laboratory at the Manned Spacecraft Center, Houston, Texas. Samples were examined, described, photographed, and weighed. They were then prepared for preliminary physical and chemical analysis. Meanwhile, a number of Earth organisms were exposed to lunar material to assure that there were no pathological hazards. Among the organisms were plants, such as conifer seedlings, and tobacco tissue cultures. Invertebrate animals such as insects, and shrimp, and the almost microscopic paramecia, and vertebrate animals such as the Japanese quail, and germ-free mice. In an intensive series of biological tests over a number of weeks, there have been no indications of pathological effects among any of the organisms. As preliminary investigations continued under the direction of a number of scientists from academic institutions and government agencies, it became apparent that 4 general types of lunar materials had been collected. There was the lunar soil itself, composed of a variety of glasses mixed with crystal and angular rock fragments. There were fine-grained crystalline volcanic rocks characterized by minerals whose crystalline faces reflected in light, and by bubbles or vesicles which formed when the rocks were molten. There were somewhat coarser-grained volcanic rocks marked by irregular cavities or vugs which formed when the rocks were partly molten. Common to both types of volcanic rocks were the whitish mineral, which is known as plagioclase feldspar, the darker one, which is clinopyroxene, and the blackish one, which is ilmenite, or an iron titanium oxide. Finally, there were the breccias, comprising fragments of volcanic rock in a matrix of lunar soil. These were formed by impact hardening. In the scientists' preliminary studies of the lunar samples in the Lunar Receiving Laboratory, there were several significant findings. For instance, all the rocks are similar chemically, which points toward a family relationship. Although the components were known, the proportions are strange, suggesting either formative processes unknown to us on Earth or an unfamiliar bulk composition for the moon. The soil and breccias contain large amounts of noble gases, that is, gases which do not combine readily in chemical reactions. Most of the gases were derived from the solar wind. In most of the rocks, there is evidence of shock. There is evidence of both lava flows and impact. Strangely, the breccias are far more magnetic than the volcanic rocks, the very material from which they were derived. The sandblasted appearance of the rocks indicates an erosion process about which there are major questions at present. There was no evidence that surface water had ever been present at the landing site. Neither was there evidence of any biological material. Perhaps the most interesting discovery was that the volcanic rocks are at least 3 billion years of age, dating back as far or perhaps further than the oldest rocks ever discovered on Earth. It would seem that if the lunar samples of the Apollo 11 landing do not themselves take us back to the origin of the moon, then certainly rocks from other regions will.
After geologists have processed the samples, portions are given to our botanical lab. They are distributed in small vials inside a sterile nitrogen environment. These vials are packed in bolt-top cans, which are transferred through a disinfectant solution into the biological enclosures used to test the plants. The contents of each vial will be placed on a plant or on tissue cultures. There is only about 1/100th of an ounce per vial. One of the most sensitive means for detecting soil-borne microbes is the seed germination test. The lunar soil sample is placed on top of seeds which have been placed in a thin chamber. Distribution of soil in this way enables us to spread a small portion over a large area. The seeds, the BR8, and the lunar soil are then watered. Within 48 hours, germination occurs for most species. Other tests are initiated in which lunar material is placed in contact with growing seedlings. This particular melon, for instance, was treated by placing 1/100th of an ounce of lunar material at the base of the plant. Watering will ensure that the lunar material contacts the young growing root tips and root hairs. A young cabbage plant is treated in a similar manner. Other plants, including lower forms such as ferns and liverworts, are likewise treated by sprinkling the lunar dust over their surface. This liverwort is growing in a nutrient solution containing various mineral salts. Another way to ensure that lunar material contacts the plant cells is to rub the leaf surface with a suspension of lunar material in either phosphate solution or water. By using a glass spatula and holding the leaf very carefully, the cells of the epidermal, or surface layer, can be gently wounded. This assures that some of the smaller particulate matter enters the cells themselves. This test is often used for detecting plant viruses. Excessive rubbing of the leaves kills them, as would be indicated by immediate wilt. Here we have the leaves of a young cantaloupe plant being rubbed with the suspension. While these operations are proceeding in the seedling cabinets, other personnel are preparing suspensions of lunar material in water for treating algae or algal cells. The suspension is made in a mineral medium known to promote the growth of a variety of algae. The small amount of lunar material is placed within the jar. After the suspension is made, it is placed on the surface of a growing culture, in this instance a green slime growing over the surface of the medium. The medium within the jar is slanted to provide a broader growing area. By shaking the suspended lunar material with the algal cell culture, both the cells and the lunar material come into contact. This assures thorough treatment. Once the suspension is made, it is returned to the flask and used as a source culture or an inoculum for a variety of other tests. The parent culture, as well as the suspension culture, are saved so that any trace of lunar material can be recovered. In a like manner, tissue cultures are treated by sprinkling with a small amount of lunar material. This is a very stringent test for both contact inhibition